Hello and welcome to the Cheeky Science Show, where in this video we're going to talk about the protein folding problem and how AlphaFold, a program developed by DeepMind, may have finally solved. And perhaps we could call it AlphaGold, considering how well it outperformed all of the other competitors at the recent CASP event, standing for Critical Assessment of Protein Structure Prediction, which is an event that happens every 2 years beginning back in 1994, whereby each competing team is given 100 different protein sequences and are given the task of being able to predict the protein structure for that protein sequence. Their predictions are then compared to the experimental model that has already been collected but not yet released to the public. Now, being able to predict a protein structure from its amino acid sequence is not a trivial task. In fact, it's been a grand challenge for more than 50 years. And this is because there are so many potential applications of having the ability to predict a protein structure, and we'll allude to some of these at the end of the video. And it's also why it's so impressive to see these results from DeepMind, that they can actually now predict a protein structure just by its amino acid sequence that, in some cases, almost matches an actual experimental model of the protein structure. So hopefully by the end of this video, you will understand why AlphaFold will change everything. And so to do this, we'll start by looking at what is the protein folding problem and give you an introduction to protein sequences and protein folding. And then we'll look at AlphaFold and the way that they make these predictions, and discuss whether structural biologists, so scientists who actually get the experimental structures of proteins using a variety of different techniques such as X-rays, NMR and cryo-EM, and discuss whether or not they're out of a job, and also talk about the next steps for AlphaFold and where next improvements need to be made. And then lastly, as I said, we'll look at the uses and applications of AlphaFold and why this is such a great achievement. So first then, what is the protein folding problem? Well, when I normally talk about proteins on this channel, I typically just draw a circle and write the protein's name inside. However, proteins don't look like that. Proteins come in all different shapes and sizes, and it can be said that the final structure of a protein can determine what that protein's function is. For example, my favourite protein, p53, has a region that enables it to bind DNA. It has a DNA binding domain. And so if another protein also had this same DNA binding domain, I would also go, maybe it's going to bind DNA. But that's the final structure of a protein. All proteins are composed of a series of building blocks referred to as amino acids. There are 20 different amino acids, and the sequence of those amino acids dictates the final structure of a protein. So the protein folding problem seeks to understand how the sequence of amino acids enables the production of the 3D structure of a protein. So the protein folding problem is under the assumption that all the information for folding the individual amino acids, otherwise referred to as the residues, is actually in the amino acid sequence. And this assumption was proved experimentally by Christian Affinson in a very nice series of experiments where he nicely demonstrates that all the information for folding residues is in the amino acid sequence. And this is well summarized in this figure here. So the protein that he was looking at is called ribonuclease, and to form the final 3D structure of ribonuclease, there are 4 bonds that are really important. And as I failed to mention earlier, it's the formation of a variety of different bonds between these different amino acids that actually enables these 3D structures of proteins to form. So what he did with this ribonuclease is that he added chemicals, in this case urea and mercaptoethanol, that caused these bonds to break, which you can see here. And this caused the structure of the protein to unravel. By then removing this urea and reoxidizing the solution, the protein structure was able to reform, as the conditions now enabled these disulfide bonds to reform, also allowing the structure to reform and restoring the activity of the ribonuclease. However, if you change the order of events such that you reoxidize the solution before urea is removed, then the protein incorrectly folds. However, this can be restored by adding a reducing agent that then enables the native structure of the protein to be formed, with the correct bonds forming between the amino acids. And so effectively what all of this does prove is that all of the information that we need is in the amino acid sequence. And so the question then is how, hence the protein folding problem. So what do we know? Well, we know that there are different forces that are driving a protein into its 3D folded structure. And the final structure often referred to as the native structure, has the lowest free energy. And so one way of kind of envisioning how this process works, this folding process, is the protein folding funnel, whereby you can see there's different routes of folding that result in the final protein structure. And it also suggests that the order in which the protein starts to fold is also important for achieving the native state. So what are these different forces that drive a protein to fold? Well, there are a variety of different forces that come into play, but one thing I want to mention is the importance of the type of amino acid that's present in the sequence. Different amino acids have different so-called R groups that contain different atoms, and this gives the amino acids individual different properties. So some amino acids have a positive charge, whilst others have a negative charge, and some have no charge at all. Well, that's never really quite true. And so as an example, an amino acid with a positive charge may be forming interactions with another amino acid with a negative charge, but these two amino acids don't have to be adjacent in the amino acid sequence. They could be much farther apart, but due to the 3D conformation of the final protein, they could be now in close proximity. Another thing that is worth mentioning is that we understand protein folding by using the different levels of protein organization. So starting with the amino acid sequence, that is often referred to as the primary structure. And this final structure here of the 3D folded protein is referred to as the tertiary structure. And so in between the primary structure, the sequence, and the tertiary structure, the final 3D structure of the protein, are, you guessed it, secondary structures. The two main secondary structures are beta strands and alpha helices. And it's understood that the series of different strands and helices within the secondary structure make up the tertiary structure. So actually, if we take a closer look at this tertiary structure here, we can actually see that it's made up of these different helices, these alpha helices, and these different beta strands, the ones that look like arrows. And it turns out that different amino acids have different propensities of forming either beta strands or alpha helices. And so already we've got some information about amino acid sequence and what the secondary structure could be. But looking at different amino acids' propensities of forming beta sheets or alpha helices is nowhere near sufficient to be able to create a 3D final structure of a protein. We need a lot more information. And one really valuable bit of information comes from the use of evolutionary covariation data. Now, that may sound complicated, but we'll get to it. Basically, this approach uses the power of comparing the protein sequence to the same protein sequence found in different organisms. For example, mice, rats, snakes, dogs, cats, whales. You get my point. And you take the same sequences from these different organisms and you form multiple sequence alignments. You see which amino acids are the same or different in the different species. Now, this is really important for identifying amino acids that are really important in the folding process. And so if we go back to that example I mentioned earlier about having a positively charged amino acid and a negatively charged one that are forming an interaction in 3D space that's important for the final structure of a protein, I can generate a hypothetical example to try and explain to you the importance of this evolutionary covariation data. So in this example sequence, let's say we've got 9 amino acids here, and one of them is this positively charged amino acid, so it could be lysine, and one of them is negatively charged, so it could be glutamate. And let's just assume, for simplicity, that the rest of the amino acids are glycine. Okay, so we've got this positively charged amino acid, and we've got this negatively charged one. Now let's compare this sequence to these different species. So we can see in this case that the dog also has the same positive and negative charged amino acid. It's got that lysine and glutamate, and it's also present in the whale. But if we look at the cat actually has them switched round. It's got the negatively charged amino acid first and the positively charged one second. And what you might notice as well is that we don't seem to have any species that have 2 positive amino acids in either of those positions or 2 negative amino acids in either of those positions, suggesting that those 2 residues show evolutionary covariation. If one of them changes, the other one has to change to maintain an interaction between those 2 positions in the final 3D structure. And so I've only really talked about 2 different features that can be extracted from the protein sequence that give us a better understanding of what the final 3D structure might look like. but there's a lot more features that could be analysed. I mean, I could be talking about it all day, but I don't think you really want that right now. But the challenge is taking all this information and being able to put it all together and try and work out what the final structure is. And so this is when machine learning techniques come into play. And so this is when we can now come back to the CASP competition, whereby different companies, different labs have generated different programs using different techniques to extract the information from a protein sequence and try and predict accurate 3D protein models. And given that this year was CASP 13, it's not a trivial process. However, as I said at the start of this video, AlphaFold has got very close, at least in some cases, as you can see in this figure here, to actually getting a very accurate prediction of a 3D structure such that it almost virtually overlaps with the actual collected experimental data. But we'll come back to that as well at the end. So how did they actually do it? Well, the first concept we have to get in our heads is to understand what they mean by a folded protein can be thought of as a spatial graph. And so effectively, if we draw a set of 3D coordinates and plonk in a protein in the middle, effectively, if you wanted to do a kind of sketch of what that protein would look like, you would need to have a 3-point coordinate to match where each amino acid will be positioned within that 3D graph. Don't know if that made any sense. And then based on the order of amino acids in a sequence, you can then just do a line that kind of connects it all up together. So in this kind of dodgy drawing I've done here, I've tried to demonstrate that I've pinpointed an alpha helix. And so if I do my line, you can see that you've got an alpha helix, and there's a beta strand in there as well. The point is that we can actually plot a protein structure onto a graph. And then the question is, what are the points of each of the amino acids? That's the information that we need. So two really important bits of information they can generate to get those positions is firstly getting the pairwise distances between residues. So if I just take this residue, for example, it's finding out what the distance is between this residue and all the different residues within the protein. And secondly, finding out what the angle is between this amino acid residue and the one either side of it, the so-called backbone torsion angles. These two bits of information are vital to being able to create a 3D structure representation. And so the latest version of AlphaFold, which they presented at CASP 14, got it right this time, involves using neural network system, which they call an attention-based neural network system, that attempts to interpret the structure of this graph while reasoning over the implicit graph that it's building. So effectively what this means is how they can use all these different features that they can extract from the protein sequence to generate these pairwise distances and angles to generate their structure. And so this is better shown if we look at their neural network model architecture. So we can see here their protein sequence, and then we can see that they perform this multiple sequence alignment that I mentioned earlier, which basically exploits the evolutionary history by comparing two different species sequences and comparing that sequence to that found in different species to identify important amino acid contact points. And then also looking at a variety of other features that enable you to calculate these pairwise distances between the residues. And although they haven't actually published a paper yet talking about their updated AlphaFold system, it seems to be that this process is iterative, such that the information is passed between these different representations to then finally generate a matrix that gives the information to form the final 3D structure. So how long does this process take? Well, if we go to a cell, it can fold a protein in a fraction of a second, so fast. And if we go to experimental models such as Xray crystallography or NMR or CryoEM, it can take months up to several years to get a final 3D structure. DeepMind's AlphaFold can do it in a couple of days. And it's not just fast compared to experimental approaches, but it's also accurate. They achieved a score of 92.4 across all of their targets at CASP 14, and so this score effectively takes into account how far each amino acid residue in their predicted model is from the actual experimental model. So does this mean that structural biologists who generate protein structures using CryoEM, Xray, and NMR are out of a job? Well, not quite. Firstly, AlphaFold was trained by using the publicly available data consisting of of around 170,000 protein structures that have been experimentally generated. So if they want to improve AlphaFold, they need to be able to at least compare it to some experimental data. And I initially thought that this might be quite a weak argument, but especially for the validation, it's actually quite important. It's important to know if the structures that they're predicting are actually correct. And secondly, Xray, crystallography and CryoEM, just the two examples, have seen massive improvements over the last few years, and they can actually generate information that can't be gained by using AlphaFold. For example, you can now get CryoEM structures of not just one protein, but entire protein complexes, how one protein is sandwiched amongst other proteins, and having a full picture of the entire complex gives you so much more information than just that protein individually, as many proteins function in a cell as a complex. A second advantage to experimental approaches is that you can get the protein on different structures. For example, going back to my favourite protein, p53, a decent number of structures have been released that not only show p53 in its tetrameric form, so it doesn't work alone, it works with 4 of its equal buddies, but it also includes DNA in the structure, so we can see how the protein is interacting with the DNA. And it's not just DNA, we can do it with RNA or other small molecules using experimental systems. And so far, AlphaFold, I don't think it can actually do any of that. It's just getting the structure of a protein based on one individual amino acid sequence. And another advantage to experimental approaches, such as using NMR, nuclear magnetic resonance, you can sometimes get quite an insight into the dynamics of a protein. Because proteins, we draw them as like static entities, but in a cell that all, do all sorts of kind of conformational changes, and that's also really important for a protein function. But yes, there are many advantages to AlphaFold. Not only the speed, as I mentioned earlier, it's kind of crazy how fast it is in comparison to some of the X-ray structures, but it's also useful for being able to generate at least predicted structures for proteins that previously were really challenging to try and crystallize. Because, the reason X-ray crystallography CryoEM takes so long is because it's so hard to get the right experimental conditions in which a protein may actually crystallize. And you may never find that condition. Some proteins may never crystallize, which is why having something like AlphaFold, where it can predict, we'll never know how accurate it is, or maybe not as accurate, but it's better than having no structure at all. And so I think this nicely brings me on to what are the uses of having protein structures. And I'm actually going to kind of split this into two. Firstly, talk about the use of having protein folding structure. Having protein folding predicting systems, the advantages of those and what we can use with that information. And then more generally looking at what we can gain from a protein structure. So let's start with the use of having something like AlphaFold. Well, what's really interesting about AlphaFold is that it collects all this different information about amino acid sequences and brings it all together to generate a 3D structure. And now one thing that would be really interesting would be to kind of reverse this process and work backwards to try and understand what information it was using, what was more important to generate the 3D structure prediction, as that might give us some information into how the folding mechanism actually works within a cell. Because that, understanding the folding mechanism is one of the aspects that goes along with trying to crack the protein folding problem. And if this could be understood, it may also then be possible to create your own protein structure and then to use the reverse mechanism to kind of see what the original protein sequence would be. But I think I'm a little bit ahead of myself. What are the actual more practical uses of AlphaFold? Well, firstly, having protein structures gives us insight into the protein's function, and that's just useful for understanding what that cell's doing and understanding what's going on within each of our cells. But being more specific, there's a variety of things that we can get with a protein structure. Firstly, it gives us an understanding of interactions, for example, with different drugs, and so this is really useful for drug discovery and therapeutics. And then secondly, a lot of diseases are caused by genetic mutations within different genes. And those genetic mutations can often alter the amino acid present within a protein. And so by having this predictive system, we can see how the protein changes when that amino acid is changed and try to understand the disease mechanism. And similarly with this, actually kind of a bit not quite related, are diseases such as Alzheimer's disease and type 2 diabetes, where it's often seen that proteins aggregate. and understanding the aggregation process can be greatly aided by having the structures of these different proteins. For example, the tau protein, which is the protein that can aggregate in Alzheimer's disease, was only recently solved by CryoEM, but is one of those proteins that was really challenging to study, and so something like AlphaFold is a really great tool. And similarly, loss of proteostasis happens in the aging process, so understanding how protein structures change during age could also be aided through AlphaFold. But my point is, there's a lot of applications of using something like AlphaFold. I mean, there wouldn't be the CASP competition to begin with if it wasn't going to be important. And so hopefully this video has given you some insights into protein folding, into the AlphaFold system, and into the potential applications. And so maybe now we can understand this quote that it will change everything, which I used as a little bit of clickbait for this video. I guess I'll see later if it actually worked. Anyway, so as always, I hope you've learned something in this video. I actually really enjoyed making this one. I got proper nerdy and into it, but I hope you've learned something. And as always, thanks for listening.